So our shoreline survey is an opportunity to collect two types of information. One, we're interested in evidence of nearby potentially green crab populations, but also other crabs that might not be attracted to our traps. We can tell if they're living nearby by looking for their molts. So Sean was talking about hairy helmet crabs never ending up in traps even though they live in these habitats. So the way we find out that they live in these habitats is by looking for their molts. Here is also where we're getting information on habitat features. So we're interested in the quality of these habitats, how it's associated with green crabs and the other organisms that live there, and how it changes over time. So this is part of your monthly survey that you can either do it on the first day or the second day of sampling. Some people find it easier to do on the first day since that setting the traps doesn't take very long at all. But to us, whatever's more convenient for you is just fine. It also occurs April through September, same deal. It consists of two components. We're gathering habitat information by setting one 50-meter transect line. This is a rope along which we're going to take subsamples to look at what the habitat is made up of. Along that line, we're going to use 10 quadrats. This is a frame of a known size, and we're going to estimate the cover of different categories of habitat inside this frame and then extrapolate out. So we're going to take multiple subsamples, 10 in this case, along the quadrat, and then we're going to look for molts in a systematic but flexible way by doing a 20-minute molt hunt. So sort of like an Easter egg hunt, treasure hunt, only we're looking for crab shells. So I'll walk through this in more detail now. So for our shoreline survey, we are interested in what kind of habitat that site provides. So we're going to set our transect line along the habitat boundary, the deepest edge of the habitat that we think crabs will be using. In a lot of cases, that will be the lower boundary at which the vegetation, the terrestrial vegetation, grows. Usually this is pickleweed, sometimes marsh grasses. Oftentimes at these sites, the lower edge of the boundary is actually pretty discrete. It might be wavy and it might be patchy, and we'll talk about that at your individual site. But in general, we're interested with what green crabs are doing at the lower edge of that habitat boundary. We also have some sites that are characterized by riprap, and so that ends up being where green crabs live, in that riprap, so we would be interested in capturing that habitat boundary. So these are the zones we're interested in, along which we set our transect line. The key here is that we will help you find this at your site. All of the sites are different in a way that if I were to try to explain every possible contingency here, we would be here well past tomorrow. So this is something that we're really going to help you find at your site and figure out what it is that your site looks like. So we've given you, as I mentioned, a pre-marked 50-meter rope marked with black marks every 1 meter. And you lay this so that it follows the contour of your habitat. You can see here that there are gaps and it's patchy, but the idea is to sort of connect the dots so you're finding the lower boundary. Now, you're going to collect cover information, what's covering the ground, at 10 random subsamples. Now, I talked about haphazard sampling. Here, I actually mean truly random, meaning we've used random number tables to assign mathematical randomness to the distances along that rope at which we collect these subsamples. So if you turn to page 9 in your big, thick handbook packet, in the upper right-hand corner, you will see a table that has lists of what look like random numbers ranging from 1 through 50. So those are the distances along which you're going to stop, put down your quadrat, and look at what is enclosed in that space. Changes every month, but it's the same at all sites for a given month. So you're walking down. What's the first distance for April for this year? 5. So you're going to walk to 5 meters from your site marker on the rope. We'll show a site set up here in a second. You're going to set down your quadrat, and you're going to record percent cover of what's in that frame. So this is what it looks like. Similar to the trapping, it's based off of the location of your permanent, semi-permanent site marker, where you'll start the rope just adjacent to it at the lower edge of the habitat boundary. And it runs along the contours, And then at each of the prescribed distances, you'll set a quadrat. Obviously, we're not giving you 10 of these things. You just take one and you walk it down sequentially. But this is just for the sake of showing you what it would look like. Now, in terms of, okay, 5 meters, what does that actually mean? Where do I set it with respect to the 5 meter mark? Because you can see these are different on either sides of the rope. That's different, very different things to look at. So first of all, set it to the positive side of the meter mark. And so if it's at 1 meter, you set it 1+. And on the shoreward or landward side of the rope. So we've put the rope on the lower boundary of the habitat. We're interested in taking observations on that habitat. So we want to put the quadrat in the actual habitat that we're interested in. So shoreward side, positive of the 1 meter mark, or the relevant meter mark. We are interested, in terms of what it is we're actually recording, in 3 broad categories. One is wrack. Looking at the things that have washed up on the shore can tell us more about the community than we're able to see just by looking at the ground. So if there is, in this case, lots of seaweeds and eelgrass washed up, we know that there's seaweeds and eelgrasses offshore that are influencing the offshore habitat. We're also interested in whether or not there are attached living organisms, epifauna. So some of these sites, especially riprap, have barnacles growing on them. These are not only organisms that make up the community, but these are potential green crab food. So if we expect green crabs to come in, if they do come in, and we see changes in barnacles and mussels that are living on our shoreline, then we might be able to attribute those to the actual predation by green crabs. So that's why we're tracking that. And then lastly, we're interested in just what the ground is made up of. How much of it is covered by vegetation that's actually rooted there versus just bare substrate, sand, dirt, rocks. So you set your quadrat down, and this is divided into 4 sections, so you know that this is roughly 25% of the area. And you can categorize the enclosed. So estimating how much area, what the percent area, sometimes you have to mentally manipulate where in the quadrat things are located. So for instance, here we have this brown patch is actually washed up eelgrass, so this is wrack. And if we imagine compiling all of this brown into one, sort of squishing it all to one side, I would suspect it would take up more than 25%, probably maybe almost half of another square, so closer to probably about 40%. You'll get a chance to practice this in groups as soon as I'm finished explaining. So in each quadrat, there are 3 steps that you go through to estimate percent cover. First, we want to look at the wrack. The wrack is anything that has washed up. It's not fixed on site. We're interested in 4 categories of wrack. There might be no wrack on your quadrat. That's fine. All of these could be 0. It might be that your site is complete, or your little quadrat is completely covered with wrack. So it might total 100%. But it breaks down into 4 categories that we're interested in. One is eelgrass. So eelgrass will not be growing as high up on the shore as we are setting these quadrats. So it will all have been ripped up and it will be in the wrack. It might still be alive because it might just have recently been uprooted. We're interested in any kind of seaweed. Again, it's not growing that high up on the shore. It's not fixed that high, but it could still be alive if it just was dropped on a recent high tide. We're interested in how much of it is land plants. So anything that is not rooted at that site but that has washed in, but ultimately it came from a tree or grass on land. So often these are just fallen leaves, depending on what time of year it is. Large woody debris, a big log, if that happened to fall right in your quadrat, that is terrestrial vegetation. So anything that comes from a land plant. And the last category is trash. Trash is anything that comes from humans, even if it's biodegradable, and even if it's a natural product. So if you see an orange peel, that counts as trash. If you see a 2x4, that counts as trash. If it's there because somebody littered it, it counts as trash. You're going to estimate percent cover of the wrack for these 4 categories. It could total, if you total up each of these 4 categories, it can range between 0 and 100. Oftentimes, rack is sort of, it occurs in layers, and if you sort of lifted it up, you might see a different kind of rack underneath. All we're interested in is the very top layer. If you look down from above and took a picture, what information would you get from that? So you don't have to dig around and dig through it to try to figure out exactly how much biomass is of each category. Estimate only to the nearest 5%. It's okay if there's 0.02% something. If it's present in such low abundances that it doesn't make 5%, just don't count it at all. So it's, we might miss very small percentages of things, but we're doing enough samples that if it's at least 5% of the shoreline, we'll capture it. Now, even though you don't disturb it while you're looking at the rack, after you have finished categorizing all the rack, you want to remove it because we're interested in the habitat that's underneath the rack. But we want you to do that first, otherwise, if you start trying to figure out what's underneath it, then you move it and you've sort of altered your sample. So remove the rack, and then we're interested in looking at what the ground is covered by or made up of. The 2 large categories that are things that might be covering the ground are live rooted vegetation. So if it is in place such that when you sort of tug on it, it's actually growing there, that counts as live rooted vegetation. The other is live attached organisms. For instance, barnacles and mussels are 2 of the most common things that you're going to see. So we want you to, how much of the quadrat is covered by live attached organisms? Everything else is considered bare substrate. We haven't come across other categories of something where, I'm curious about some contingencies, but for instance, if you come across a dead organism, a crab molt, we're not counting it in this part of the survey at all, as it would be considered bare substrate. This includes a dead fish. If you come across a dead fish, it's, even though it might have floated in there, we're not counting it in our rack.
So bare substrate is like
sand, rocks, mud, anything that's not alive. Yeah, exactly. So for instance, this is the most common type of live rooted vegetation. This is actually in Padilla Bay. I know where this is, just up by the gun club. Which gun club? There's a million. Sullivan Minor. This is bare substrate for the most part. You can see there's probably small patches here of either washed up seaweed or eelgrass. And live attached organisms. I just put this here because we didn't collect this last year, but we decided it's something we're really interested in. Usually mussels and barnacles, we would count, in this case, limpets or snails as well. So live attached organisms, I want to take a second. Probably I don't have to tell you, most of you, that you can tell if a mussel is alive, not because it's singing or anything, but because it's clamped shut. When mussels die, their adductor muscles relax and allow it to gape, and so if there's a gaping mussel, it's dead. If it's open at all and if it doesn't respond if you flick it, then it's dead. Sometimes if mussels are heat stressed when the tide is low, they do open up a little and allow evaporation to cool them, because you can imagine sitting in a black shell on a sunny low tide in the middle of the day, you start to steam yourself, literally. So they will do that, but if you flick them, they should close up in response to that if they're still living. Barnacles there's also a sort of general rule of thumb. I know about people who like whisper to barnacles and poke them with very small things, and we're not going to ask you to do that to individual barnacles. What we're asking you to do is look at whether or not the barnacle outer shell, which is called a test, is empty, in which case it's dead, or if it's full. If you see these opercular plates, the plates that cover their opening and that cover the organism living inside, then typically that means the barnacle is still in there. So that's our rule of thumb. So for estimating, you can see it could be a little bit complex because there's some interspersed living and dead ones here, but how much of the area is covered by the dead ones versus the living ones. And keeping in mind that the whole barnacle extends all the way around. It's not just this tiny little inset hole, it's the footprint of the barnacle that you're really interested in. Okay. Yeah, so by proportion, not by number of individuals. Yeah, percent. So 25% of the area is dead barnacles, so that's actually bare substrate. So again, going back to just living things are considered not bare substrate, plants and animals. The last thing we're interested in, step number 3, is dominant substrate type. This is an estimate of the grain size, and you can get really technical, with a pile of sieves and shaking it and sorting out the profile of sediment sizes. We're not going to ask you to do that. We're just asking you for sort of a broad brush approximation of, is it muddy, is it sandy, is it gravelly, is it cobbly? And for me, the distinction between gravel and cobble is, would you use it on your driveway? You would not use, for instance, anything larger than a softball to pave your driveway. But anything that you feel reasonably sure would be okay on a driveway, I would call that gravel. There are some riprap sites, so the substrate might actually be riprap, in which case, of course, it will be human constructed. And then lastly, we don't expect anybody to have bedrock sites because those are high energy sites, but we put it there for the sake of completeness. In case there's maybe one spot at your beach, but in all likelihood, nobody should be using that category. So this is the data sheet. You'll notice the shoreline survey data sheet has a front and a back. This is the front, which has your quadrat data on it. There's a separate chunk for each quadrat that walks you through each of the 3 categories. So here you have your metadata. Note that there's no spot for hours on this sheet. Your hours should be included in your trapping data sheet, and that should cover this part of the survey as well. Now, the first thing you do is you write down the random distance that quadrat 1 was at. This is probably from last year's, which is why it's 8 instead of 5. But whatever distance, however many meters you went to get the first one, that's what you put there. Then step 1 within your quadrat is estimate the rack and then remove. So how much of the rack is eelgrass, how much is terrestrial vegetation, that woody debris, seaweed, and trash. Again, could range in the total from 0 to 100. It should not go over 100 because you're just looking at the top layer. Number 2, we look at cover. How much bare substrate, live rooted vegetation, and live epifauna do we have? This should total exactly 100%, no more, no less. And then lastly, we select our sediment type. Again, circle only one of these categories to give the dominant substrate type. If you have, for instance, a few cobbles in a muddy area, whichever one takes up more space, that's the substrate type you choose. And then we can move on to our next quadrat and sort of proceed sequentially 1, 2, 3, 4, etc. Okay, any questions about that? We'll get a chance to practice it briefly at least. Okay. The next thing that we're going to do is look for molt. So we're looking for evidence of nearby crustacean populations. Well, we might see shrimp molts, but mostly crabs. So here we're doing a timed survey where we want to collect all of the molts that we can find, everything we can find, and spend exactly 20 volunteer minutes doing this. So you could do 2 volunteers for 10 minutes, 1 person for 20 minutes, 4 people for 5 minutes each, right? Math. Count and identify the molts after the time is up. So you're just collecting them, and then you look at the spoils of what you got. We don't want you to have to spend collection time trying to figure out what it is you're looking at. We want you to actively target areas where molts are really dense. So we're trying to find as many crabs as we possibly can, knowing that this is a way that we could find green crabs. So the more molts we find, the more likely it is that we would stumble on green crab molts if they are there.
How do we determine the area we're looking at?
Sure, I'll show you a little bit a schematic of that in just the next slide, I think. The areas where molts tend to accumulate, certainly the rack line has a lot of molts in it, regardless of how high it is. If you're there on a low tide series, the rack line is actually probably fairly high up on the shore. But there might be also multiple rack lines, and you can look at as many rack lines as there are. There also tends to be patchy distribution of molts. You'll get familiar with your site, and we can talk about it the first time that we go out with you. So think of this as a molt Easter egg hunt. I typed in like crab Easter egg hunt to Google searches, and I don't know if anybody heard about this, but Walmart was selling these plastic Easter eggs, and it turned out that some lady in hers found actual crabs in like actual crab shells, bits of dead crab. I think it's crab shell. I don't think it was actually a dead animal. So in any case, I don't expect you will actually find this in the tide flat. But in any case, think about it. Think about it this way. It's a treasure hunt, and you're trying to find the most mults possible. So this is getting to your question about where to look. So the way we think about it is basically anything at your site is fair game. But in order to try to standardize it so that even at these very different sites, we're kind of giving every molt a fair shot of being found, we'd like you to start by standing, whoever is participating in the molt survey, stand at the site marker and visually scan for areas of high molt density. And they could be on either side of your stake. So if it turns out you actually turn around, your traps are behind you, you turn around and there's a big patch of mults there, go for it. Don't feel limited by the area where your trap string is set up. We're really just trying to be sensitive to find as many mults as possible. So here we have areas in the rack line where there's a lot, and sometimes they accumulate along that bottom edge of vegetation. They get tangled up as the tide comes in. So you and your partner, say you're doing it with someone, I mean your competition, because obviously this is a race, are standing there scoping out sites of high molt density. Start an actual timer so that you don't have to keep checking your watch. That will go off when it hits 10 minutes or 20 minutes, depending on how many people you have. And then you can use the bin for this purpose, or if you have any other bucket. They took my bins, so I'll use a pretend one. And go to the sites where you see the mults are very dense. And this is a little bit wonky, but what we want you to do is, so first, the way you allocate the time, as I mentioned, you can do it in this way. I think doing it as 10 minutes for 2 people is probably the best way. If you have a ton of people out on the beach, then you have a lot of interference with people jostling you for mults. We'd rather that not play a factor in the amount of time that people spend capturing mults. And then if you're just one person doing it for 20 minutes, it can get kind of boring. So a little friendly competition, I think, is healthy. So then the sort of nitpicky thing here is that we'd like you to place the mults in the bin once at a time. And the reason for this is you can imagine if you've seen piles of mults washed up on the shore, that the temptation might be to hold your bin and then just sweep about 1,000 mults into your bin. But then you got to sit down and count all those mults, and that doesn't seem like very much fun. So in order to put sort of like a maximum rate at which you could collect mults, and it sounds silly, you might just want to pick them up and do this. It's very tempting, but just do them one at a time. The other thing is that you will have noticed handling these mults that they're kind of fragile. So if you just kind of take a fistful and put them in there, suddenly all you have is molt dust. It's kind of hard to identify molt dust. So this is just a little reminder of doing it one at a time. You don't have to be exaggeratedly slow about this. This is still a competition, but it has to be a fair competition. Okay, once you have your timer goes off, take all of your bins and you can put them together and count and identify the mults. Now the key here you'll notice that mults often tend to be broken up into pieces, and when they look like this, sometimes they're hard to identify. You might be able to look at the claws and tell what they are, but there's this other tricky bit here, which is this bottom of the crab and claw might have actually come from this crab. If you count this and this, you'll predict that there are 2 crabs there when actually it all came from one. So if you find a partial molt like this, we don't want you to even bother trying to identify it. You don't have to collect it if you see it and there's no carapace. However, if you have even 50% or greater of the carapace, that's what we'd like you to count. This keeps us from double counting, and the carapace will give you all of the information you need. Here, anyone tell me what this one is? Look at these blunt teeth. Red rock crab, good. So we've got a pointy rostral shelf, and blunt teeth. And we have just over 50% of this carapace. So that's how you tell which ones you want to count. The data for this is on the back side of that shoreline survey. It's fairly straightforward. Mostly every species just gets its own row, and then you can use this space to tally as you go, so you can total multiple species at the same time. So, have somebody going through the bin saying, he knew, he/or, CAGR, CAPR. CAGR is actually not a crab on here. This is a long story. But so if someone says that, you should tell them to take a second and double check what it is. And then totaling them here on the edge. So this is essentially all. We're not asking for pictures. We're not asking for sizes or sexes. This is a very simple tally count of how many molts are out there and what species are they. If you find a green crab molt, same protocol for calling Jeff in the middle of the night and giving him a hard time and demanding that he come down and help you immediately, wherever he is. You can keep the molt. You can still freeze it in a Ziploc bag. Sometimes they get debris in there that gets kind of stinky. It's up to you. You don't have to. If you can't identify a species, the reason you can't identify it is because you don't have enough of the shell to find the features, then you don't count it at all. You can keep the molt. If you just can't tell what it is because you can't tell what it is, you've never seen it before, you can go ahead and keep the molt. It's not a live creature. You're not doing any real damage to the ecosystem. Again, using a placeholder and getting an email to us with photos that include scale and some sort of description. We can get the actual molt from you if we're just like, oh my God, I have no idea what this is. We have very talented crab people on our campus. The people who write the books that we've put out there in the back are on campus, so we can ask them, what on earth is this? And so if you try to do that to us before you submit the data sheet so that we can get the answer to you, and the data sheet that you submit is therefore complete, it keeps us from having to track down, shoot, I know at Penn Cove there was some unidentified species, but was it this picture or this one? So it's best if you do all this legwork, and there probably won't be much of it. It'll be pretty rare. Before you submit your data sheet, that makes it a lot less likely that we'll make a mistake in where the data go. So this is the same situation where you submit your data after your sheet's complete by sending us an image of the one picture for the front and one picture for the back. They have these naming conventions which are in your handbook, and mailing the original data sheets at the end of the season. Do you guys have any questions about that? That's actually the whole thing. Treasure hunt. Yeah, sorry, go ahead, Bob.
Go back a little more. Well, I was wondering where that Awesome Bay is.
Oh yeah, right. I want that site. Awesome Bay is unfortunately not a real site that we have. There is no site 302. It's a joke. I know. I've sold it so heavily, and now I have to.
So if we want to put this slide up,
Yeah.
what do you want us to capture in the notes
You
here?
know, I actually haven't really thought of anything that is. If you thought it was a really weird color or something, I haven't thought of very many things, but it's always a good idea to leave a little bit of space for notes, and that is a little bit of space. It's not very much, but yeah. Don't be intimidated by this, and if you feel like you need to take space at the top of the form to write me a note, take notes in a notebook and email me. That stuff I can track down. Thanks for asking.
Do you want full name or
No, just the list of who was present. So yeah, if you want to just do initials, just enough to identify who they were. So if there's two Bobs, for instance, on the same team, maybe a last initial would be helpful. Great. I think we are all set to follow Kate out. What you need with you this time is the Shoreline Survey data sheet, your handy dandy crab ID guide, and a clipboard and pen, and that should do it.